series. We appreciate your continued participation and engagement in this informative journey organized by Mindray India for the benefit of our IVD community. Today, the lecture will focus on examination processes and release of test results, a crucial component in ensuring the accuracy and reliability of lab results. Once again, we welcome our esteemed speaker, Dr. Jayesh Warade. His extensive experience as a consultant biochemist and molecular biologist, Dr. Warade is uniquely qualified to guide us through the intricacies of the processes, sharing practical insights and the best practices. So welcome, Dr. Warade, and without wasting further time, we start at dot 7 today. So Dr. Jayesh Warade gets more of the time and also a proper discussion happens. Thank you, everyone. Thank you, Dr. Jayesh. The dais is all yours.
Yes, yes. Thank you. So I will just share the screen. So I welcome you all on this fourth session of this good clinical laboratory practices. Now, as we have seen today, there are two important topics: examination processes and release of test results. So, as from the first day I'm seeing, we have some doubts with respect to the examination processes, QC practices, validations, verifications. So basically, let us see, we will be able to solve all those queries today in the today's session. So to begin with, first of all is the examination processes. Here, as per these guidelines and even as per the international guidelines, the laboratory should select examination processes which have been verified or validated for their intended use. So it has been emphasized in each and every guidelines and in each and every standard that whatever the examination processes the laboratory is using for performing the examinations, performing the test, must be appropriately verified and validated. Okay, so it is mandatory that the laboratory must select the tests which are validated and verified. Apart from that, the identity of the person performing activities in examination processes shall be recorded. So whoever has performed the examination processes, at least the key activities, whoever has performed, their identity must be recorded. That means who has performed the examination. So suppose if you are performing any test or any examination related to histopathology, so there are multiple stages. If you see in histopathology, it is a most of the time performed by a manual examinations and it involves lot of manual steps. So there will be grossing, there will be fixation, there will be cutting, then there will be staining, and then there is the examination. So whoever has performed all these key aspects, so maybe XYZ person has performed the grossing, then maybe ABC person, somebody has performed the cutting, then there was maybe before that there was fixing, then there was cutting, so maybe some person has in between performed the staining, so again record identity that this person has done the staining, and then maybe finally whoever has performed the microscopic examination, obviously that identity. So whatever the key processes are there, all those key processes must be recorded. The identity of the persons who has performed these key activities, like there should be some traceability. Whatever activities we are performing as a part of examination, we must record the identification or identity of those persons whoever has performed those activities. Apart from that, examination procedure should be documented. Now, examination procedure means standard operating procedure. So standard operating procedures for each and every test, each and every examination should be documented. Now, in the previous lecture also itself, I have mentioned why it should be documented, so that you will be able to train all your personnel, all your staff will be trained on those SOP standard operating procedures, and whenever whoever is performing a particular test, maybe some ABC person, XYZ person, today somebody has performed, tomorrow somebody else has performed, day after tomorrow somebody else has performed. So the consistency remains the same. So if there is a in a standard operating procedure, if it is mentioned that you have to incubate for 10 minutes. So if today I am performing the examination, I will wait for 10 minutes. Tomorrow somebody else is coming, he will also wait for 10 minutes. Day after tomorrow somebody else is coming, so they will also wait for 10 minutes. So the consistency remains throughout the your examination procedures, and whatever activities you are performing, it remains same. So there should be appropriate examination procedure. Whatever is the simplest action is there, you need to have appropriate examination procedure which should be documented. And next part, written in a language commonly understood by the staff in the laboratory and be available in the appropriate location. Now, understood by the staff in the laboratory means standard operating procedure should be written in a very simple language. In a very simple language. Why? Because these are examination procedures which are mostly used by the actual workforce of the laboratory, and not necessary all this workforce is highly educated or is highly proficient in your English language or maybe any other very sophisticated language. So what you have to do, you have to write it in a very simple language so that each and everybody working in the laboratory should be able to understand those procedures. And if they are understanding each and every step, then only they will be performing the procedures or activities as per the standard operating procedure. So it is recommended that all the SOPs should be written in a very simple language and should be understood by the all the staff of the laboratory. SOPs helps to ensure uniformity, consistency, compliance with regulations, efficiency, quality output, and control over the process. Now, why this SOP should be there? As one of the things I have mentioned is there should be consistency, uniformity. So, whichever activities we are performing, it should be uniform, it should be consistent. Apart from that, compliance with the regulation. So, performing any activities, there should be a standard operating procedure. So, it is a one of the requirements. So, to maintain the compliance with those requirements, you should have a standard SOP, standard operating procedure. Efficiency. So, it will also make a more efficient. Not necessary every time you have to go and read those SOPs or you have to see those SOPs or you need to have your separate own protocol. So it will create a lot of confusion and it may decrease the overall efficiency of the laboratory. So to have a quality output, to have an efficient working condition, you must have a appropriate standard operating procedure and you will have control over the process. So there will not be any non-conformances or there will not be any deviations in any of your processes. So that purpose you should have always a standard operating procedure for even for the simplest of the activity in your laboratory, and most important part to be written in a very simple language. The laboratory personnel must be well trained in the SOPs. Again, it has been emphasized that all the personnel must be well trained in the SOPs. Well trained means what? Do they need to just learn it by heart and do they need to memorize everything? Not necessary. They need not have to memorize all those big SOPs or they need not have to learn it by heart. They should be trained, at least they should understand it and they should be work as per those standard operating procedure. They should know the importance of each and every step. Why they have to incubate for 10 minutes, why they cannot complete the examination in 5 minutes, or why they cannot terminate the procedure before that. So they should know why it is important to incubate and to wait for 10 minutes or 15 minutes, why it is necessary to perform each and every activity one after another in a step-by-step manner. So they should be trained on all the SOPs. They should know the importance of each and every step and each and every activity of the SOP. As I mentioned, it is not at all mandatory that they should memorize those SOPs or they should be learning those SOP by heart, so that you will take some written examinations and if they make some mistake, you will deduct their certain scores or certain like that. It is not like that. It is expected that they should understand each and every part of the each and every step of the standard operating procedures. Now coming to the examination processes, quality planning is concerned with establishing and validating processes that meet customer needs. So whatever plans you have, whatever processes you have in your laboratory, you must validate those processes. Validate those processes means, now I will tell you two different things are there. One is validation of the examination procedure and one is the validation of the processes. Processes means the complete activity to perform each and every individual test. It may includes multiple activities linked with each other. So validating processes means a simple activity is there, transportation of the samples. So how much is the optimum time which is required? How to maintain the transportation procedure? Are those procedures and all those processes, those conditions of transportation appropriate? Is it guaranteed that it will maintain the integrity of the samples? So for that purpose, you must have validation of all those procedures and processes so that you will find out that you will be able to optimize those processes and there will be no further deviations in any of those activities. So it is required to validate processes so that you will have a uniformity while performing any of those processes. Okay. So coming to the and verification part of the, let me see if we have no. So I think we can cover the validation of examination processes part here, because since first day I am seeing there are certain questions regarding validations and verifications. Actually, I wanted to take since we are here in the examination procedures, we will take it now itself. Now, there is a major difference. What is meant by validation of examination processes and what is meant by verification of the processes. And whether in laboratory you want to do validations or whether you want to do verifications or you want to do both. So, as the standard or as these guidelines mention that you should use only validated and verified examination processes. So basically, when does you have to do validations and when does you have to do verifications? So many are these both the terminologies interchanging, but there is a lot of difference. Verifications means the processes or the examination procedure which is already validated. The examination procedure which is already validated, that you need to verify in your laboratory. And the examination procedure which is newly developed, newly introduced, which is certain panelites or certain test parameters are newly introduced. When you introduce these new parameters, when you introduce this new procedure, that time you have to perform a validation. Suppose commonly what I am doing, I am using a test method of glucose estimation by GOD-POD method. Very commonly used, everybody is using, and I think everybody is aware of this simple process of glucose estimation by GOD-POD. So everybody is well versed with this procedure. It is quite often validated and it is validated since long. So there is no challenge. But when I want to start this glucose estimation in my laboratory from XYZ manufacturer, what I need to do? The method is already developed. The kit is already available, commercially available, kit is already available. But now what I want to do? I want to start this test in my laboratory. So I will perform the verification because it is already well known examination process, well known test method is very common. Everything is there. So I just need to verify those examination processes. So whatever claims have been mentioned by the manufacturer regarding the linearity, regarding the accuracy, regarding the precision, regarding the sensitivity, specificity, all those claims, I have to just in my laboratory. So whatever claim has been mentioned by the manufacturer, whether all of them are true or is there any deviation? Am I able to reproduce all those claims in my laboratory? Is it possible to reproduce those claims in my, what I will do? I will perform a verification. Whatever claims have been given by the manufacturer, I will just verify those claims in my laboratory. So is it clear? This is the verification. Now when it comes to validation, now what I am doing? Previously, or I was performing the glucose estimation is well validated. So when I was trying to use those examination method in my laboratory, I will perform a verification. So that is verification. Now I am working in my laboratory and by doing some examinations, by doing some research, by reading some literature, I have developed a new method for glucose estimation. Say some name, ABC examination, glucose estimation by ABC method. Now this ABC method is completely new. This ABC method is completely new. First time I have done some research and I have developed this new methodology. Then what is the sensitivity of this method? I don't know. What is the specificity? I don't know. What is the measuring range? I don't know. What is the accuracy, precision? Nothing. I have just developed that this method ABC can estimate the blood glucose in your laboratory. So now I want to establish the claims. I want to know the sensitivity of this method. I want to know the accuracy of this method which I have developed. I want to know the precision of this method. I want to know what is the linearity, what is the interferences, what can be the other applications of this test, how far it will be reliable, what type of samples is required, whether it required blood samples, whether it required any specific serum samples, or whether it requires any specific plasma samples. So all these I need to establish. So for that purpose, I am going to do a validation, because this is a newly developed test. There are no claims from anybody. First time I have invented, and now I want to perform the validation. Now you are clear what you are doing in your laboratory. Is anybody doing this validation? Are you doing any? Are you developing any new methodologies in your laboratory? So most of the time, 99% of the time, we are not developing any new method in our laboratory. We are using those methods which are IVD approved, which are well validated. We are just using those methodologies in our laboratory. So we need not have to perform any validation. 99% we are using already validated methods. So only thing you have to do in your laboratory is the verification. Apart from that, if you are developing any new method, then you will require to perform a validation. So in short, verification is confirming the claims made by the manufacturer, and in validation, in short, will defining those claims, defining the claims or defining the specifications for any test method. You have to define it by performing object or giving objective evidences. You have to come to some conclusion. What will be the specificity? What will be the sensitivity? So this is the difference between validation and verification. 99% of the time we are performing only verifications in our laboratory. Many a times, if you are attached or if you are working in some research laboratories, you are developing new tests, then you will require validation. Apart from that, if you are making some changes in the already validated test, so one test XYZ is available, or maybe we'll take same example from glucose estimation. Now this glucose estimation is a well validated for performing glucose estimation in urine, in CSF, and in blood samples. Glucose estimation can be performed by GOD-POD method in your urine samples, in your CSF samples, and in your blood samples. Now, same test GOD-POD method for glucose estimation, I want to use for glucose estimation in pleural fluid, or I want to use same test for estimation of glucose in pericardial fluid, in ascitic fluid, in synovial fluid. Now what? I am going to do something which is not recommended by manufacturer, because manufacturer has given already recommendation. This test can be used for urine, blood, and CSF. But now I want to use it. I want to try for synovial fluid. So now again I want to perform a validation. The test method remains same, but the specimen type has changed. So you have to perform the validation. You have to perform the validation, because you are extending or you are going beyond the manufacturer's claim. So that means you are doing something, you are going to try something new, you are going to try some new specimens. So you have to perform the validation, because you are going beyond the scope what is mentioned by your manufacturer. So this is second case. First case is when you are doing developing any new method in laboratory. Second case, when you are changing the, or whenever you are going to use the test method beyond the manufacturer's recommendation. So this is the second part. The third part, when you want to do a validation, is whenever you are changing the methodology. You are changing the methodology, or you are changing the procedure which is given by manufacturer. Now same for GOD-POD estimation, I am going to take for GOD-POD estimation, for estimation of blood glucose, I am going to use, or I am going to incubate for 15 minutes. It is routine protocol. Most of the time we are following 15 minutes. Now, what I have done, By over the period of time with my experience, what I have found out, even if I incubate it only for 10 minutes, it should give the correct result. So what I am doing, I am changing the protocols which is given by the manufacturer, or I am changing the protocol which is already validated. So now if I am changing the protocols, if I am changing the incubation period, or if I am changing the reagent volume, if I am changing the sample volume, if I am making any changes to this, what again I have to perform the validation, because I am going beyond the procedure whatever recommended by the manufacturer. So I am to perform the validation in this aspect. So three conditions I have told you when you have to perform the validation. And verification, yes, everybody has to do when starting a new test in your laboratory. So if you want to start thyroid test in your laboratory, T3, T4, TSH, which is well validated, but for the first time you are introducing in your laboratory, start with the verification. Verification, everybody has to do. Validations, there are only three conditions when you have to perform the validation. We are clear with verification and validation? That's it. Now, as we have seen in examination procedure, applies to selection and evaluation of new methods and instruments, as well as selection design of QC procedure. So all your examination procedures also applies standardization of your selection and evaluation of new method whenever you are starting a new test in your laboratory, or you want to upgrade the technology, you want to upgrade the methodology in your laboratory. You must evaluate the new equipment, you must evaluate the new methods, you must select and design appropriate QC procedure. You must select and design appropriate QC procedures which are required to have a appropriate validity of examination result. Now coming to the examination procedure part, the test procedure should include the following but not limited to. So examination procedures means I already mentioned these are the standard operating procedure, should be written in a very simple language, and your staff must be trained in all the standard operating procedure. So what are the important things you should include in the SOPs? So here we have certain recommendations. What are the minimum criteria or minimum important points we should be including in our SOPs? Name of the test should be included. Scope of the test. Scope of the test is for what purpose the test will be utilized. Purpose of examination. What will be the application of those tests? Principle means maybe a basic chemistry behind that. Method of procedure. Method means step by step whatever the protocol you are going to follow. Limit of detection, that is examination sensitivity. Analytical measurement range, that is AMR. Specimen type, sample collection, processing and storage of specimens. Next is reagents, which reagents are required, which equipments are required, glasswares and other accessories also should be included in the SOPs. Procedural steps, calculations, we have to perform many times certain calculations to come to a correct result. So those calculations, QC procedure, what specific QC procedure you are going to follow, how many times you are going to run the QCs, what will be your acceptable criteria for QCs, what will be your rejection criteria, all those should be included in your procedure. Interferences, what other substances which can interfere in your examination processes, such as hemoglobin levels or hemolysis, icterus, that is bilirubin concentration, lipemia, like triglyceride, high triglyceride concentration, or any other drugs, any other environmental factors which can interfere in your examination procedure. So all those interferences. Then biological reference range. Again, biological reference range means the reference interval you are going to use in your laboratory. Clinical significance, inference means conclusion, and limitations of the test. What are the limitations of this test? Those also you have to include in standard operating procedures. Critical alert, for some analyte there will be critical alert, critical results, above which or below which the clinician has to take decisions. So those critical alerts also you have to include in your examination processes. Hazardous material, so this, as I have already mentioned, you should be knowing about what are the composition of those reagents. There will be certain acids, there will be certain alkalis in that reagent may be available. certain hazardous chemicals will be there, certain carcinogenic chemicals will be there. So how to handle those hazardous material? What are the precautions and safety precautions must be taken while handling and while storing those reagents and even while discarding those materials? Limitations and potential sources of the variation. What can be the sources of variation? Timing of the sample collection, transportation of the sample collection, various preservatives, they can limit and they can cause potential variations in your results. And finally, the references. Now, references means from wherever you have taken that examination method or from wherever you have taken that standard operating procedure, from wherever you have collected this data, a proper reference should be available. Now, many a times what we have seen, when you have to prepare the SOPs, what we will do, we will just take the kit literature PDF copy, we will just copy and paste. But as I have mentioned, it should be understood by your technical staff. And if you have seen these all kit literatures or kit insert, they are written in a many times in a quite complicated English language, or there are maybe some complex English wordings is there. So it may not be possible for your all technical staff to understand those meanings. So that is why just copy and pasting the kit insert is not going to solve your purpose. You have to prepare your SOPs in a very simple language. You should know what is relevant part for your technical staff, what is not relevant for them, what areas they should emphasize. For that purpose, you should always have a customized standard operating procedure and it should not be the copy paste of your kit insert. Now, coming to the performance characteristic of a method, as I have mentioned, these performance characteristics includes your precision, accuracy, range, analytical measurement range, that is what is the lower limit and what is the higher limit your equipment can measure or your test examination can reliably measure. Clinical reportable range. Now, clinical reportable range may vary from your analytical measurement range. Why? Because many times, if the values are more than analytical measurement range, we have to dilute the sample with suitable diluent. The sample should be diluted by suitable diluent. Now, even the dilution can be performed with the help of distilled water, normal saline, or only by the diluent which is recommended by the manufacturer. So whatever is the recommended diluent is there, those diluents should be used whenever you are getting the values or whenever you are getting the results above your analytical measurement range. So this will be called as a clinically reportable range. So here it is mentioned that when sample is diluted to report higher values does not covered by AMR. So your AMR may be very at low level, but after dilution you can report even at higher values or even higher values can be reported or result can be reported by your laboratory. For that purpose, you should have a specific clinical reportable range. How to come to the conclusion of this clinical reporting range? For that purpose, you have to perform certain dilution studies or linearity studies. To what level, up to what dilution the relation between the dilution values and the results obtained remains the in a linear relation, maybe direct relation or maybe inverse relation. So those relations you can verify with the help of your dilution studies to come to appropriate clinically reportable range. Next is the biological reference interval. This is also one of the performance characteristic of a method because depending upon the test method, the biological reference interval may vary. Depending upon the population you are working or depending upon your geographical location, the biological reference interval may vary. Depending upon the age, depending upon the sex, the biological reference intervals may vary. So you should define the biological reference interval depending upon the geographical area, depending upon the test methodology you are using, and depending upon the age and sex factors of the population. Then analytical sensitivity, as I have already mentioned during my first lecture, analytical sensitivity is basically the lowest concentration which a test examination can detect. Analytical specificity is basically the Analyte of interest only should be detected. There should not be any interference or there should not be any mix up from any other analyte or from any other chemicals while performing the examination method. So that is analytical specificity. So this was related to the examination part. Now coming to the release of test result. So again, a sample test report format is given in this good clinical laboratory practices. I will show it you here. Before that, I will tell you what are the components it should be included in the test report format. Name and unique patient identification number. Both the things should be included. Date and time of specimen collection. Now, many a times I have seen some laboratories do issue the report, but there is no mention of the time of collection, there is no mention of the date of the sample collection, and most of the time only some arbitrary one date is given. Now I am not sure this date is about sample collection or whether this date is of report release. So you should mention specifically date and time of sample collection, date and time of test done and result reported. So when the test was done, when the test was completed, and when the result was reported, all this date and time must be included in your report format. Name and address of the laboratory, again it is one of the important part, because there can be multiple laboratories with the same name, XYZ laboratory, in one corner of the same city, in another corner of the same city. There are some names which are very common and used very by many laboratories, or maybe many a times there are some certain chain of laboratories which works or which functions in multiple cities, multiple parts of the same cities. So name and complete address of the laboratory wherever the tests have been performed must be available in your test report. Name and source of the sample. So what was the sample? Blood sample, urine samples, sputum samples, CSF samples. Name of the sample should be available, and also source of sample. This also I have mentioned during my last class. Source means basically from wherever you have collected the samples, whether it is any particular lymph node groups, or whether it is any particular section of arm or forearm or whatever part, or if they have taken collected some swabs, then you should know the swab location also. Many a times I have seen some microbiological reports. Specifically, I will give an example. We are getting reports, or we are seeing some reports of OT swabs are there. It is required many a times now for accreditation purpose and certification purpose regarding the sterility of your OT and regarding the sterility of your labor rooms or sterility of your ICU. So we collect the swabs and we give to some laboratories and they give us report. The culture is sterile. The swab, no growth is found. But from where you have taken the swab? OT. Which OT? Maybe in some big hospital there will be 3-4 OTs. So make a specific location of the OT. Even from OT, from where you have taken the swab? Maybe from the OT table, you have taken from crash cart, or maybe you have taken from some other location. So make a specific location, write down the specific source of sample from wherever the sample is collected. So this is the format which I was talking regarding the sample reporting. So here it is given. Now these are all the sample format. You need not have to have like this, because if you see this is a very rough format, or you can see there is no, it is not at all attractive, or just some A4 sheet is thing. You can think of some A4 sheet, ruled A4 sheet where something is written. So the component should be included. The format, the design you can include as whatever is up to your choice. So name of the laboratory, name of the patient, age, sex of the patient, OPD, UID number, referred by whoever has referred the case. Now, why you want to include the case? Because whenever somebody else is referring the examination result, they should know who has ordered the test. Maybe the patient themselves have ordered, or some clinician or some doctor has referred this case. Then date of specimen collection, date and time of specimen receipt, date and time of report, name of the test, method. This is very important, because I have already mentioned reference ranges many a times depend upon the test method which is used. Then test results. What is your reference interval, biological reference interval? What is interpretation? Now this is not required for all the tests. Maybe a simple blood glucose test is performed. You may not require to put all those long interpretations. You can just mark it high or low, that is sufficient. If it is within reference range, you need not have to mark it. But if it is critical high, critical low, if it is abnormal, preferably try to give it with some color code so that it itself will be a self-explanatory. You may not have to put a interpretation, but there are certain tests where you have to perform an appropriate interpretation. So in that cases, you may require to put interpretation. Then date of reporting, whoever has reported, or date of what time or on what date the report has been issued, and next is the finally the authorized signatory. Authorized signatory means whoever have reviewed and reported the test result, that person must sign the report of the result. Now coming to the release of test results, again the test results method, measurement units, biological reference interval, clinical decision units or cutoff values where it is applicable must be included. It must be signed by the authorized signatory. So all these components should be included as a part of release of your test result. Now next is the authorization for amendment procedure should be specified in the SOPs. Amendment procedure here means if sometimes in case you want to amend some result or maybe one report you have already released and now you want to make some changes, so there should be a procedure how you are going to make the changes in the result which is already released or which is already reported. So you must have appropriate procedure, and for that only authorized personnel should be able to change those report or should be able to edit those report. Not everybody, each and everybody should be able or should be given right to edit the report. So you must have appropriate procedure by which you should be able to have a procedure in place so that whenever you require to make any changes to the report or whenever you want to edit, many times we have seen, we have written some report, most of the time for the histopathological cases we are typed the report, the authorized signatory has signed it, and after that somebody remembers that oh there was some spelling mistake, there was some spelling mistake, now you have to correct that spelling mistake. So again, even if only you want to make some correction in the spelling mistake, let it be a authorization. Not everybody should be able to make those amendments. Somebody who has proper authority, they should make the changes to that report and they should amend the report. Many times there are some serious consequences. Again, for histopathology, why I am taking histopathology? Because most of the time there are manual examinations and the reports are transcribed manually. So most of the time chances are there that where you have to make some amendment to the report. Like I have received some biopsy of the right ovary, and then I have, with the help of those specimen, I have just mentioned that it is something carcinoma of the ovary, some type of XYZ carcinoma of the ovary, and the specimen which was received was the right ovary. But while typing, instead of right, the typist might have type it as a left ovary. Now, what is the consequences? Unnecessary. The clinician has to explore and remove the left ovary also. So, you should know the consequences of the reports whichever you are releasing. For that purpose, only authorized personnel should be able to edit or amend those reports in your laboratory. So, you must have appropriate procedure for amendment. All amended reports should be released with an appropriate comment. Appropriate comment means you should mention in the comment whatever is the reason. Don't shy. Don't feel that what patient will think, what doctor will think, whether they will go or come and fight with us. Don't shy away. Whatever is reason, whatever was, even if it was a spelling mistake or some identification mistake, just put it in your comment and make it a document as a evidence. The laboratory should also record the reason for editing the data. Now, whenever you are changing, make it a reason. Record the reason why you are changing, why you are going to amend the examination, why you are going to change the report. So, you should not shy away by giving appropriate comments and appropriate reasons for amending any of the test results. Laboratory should maintain a list of tests with critical value. So your laboratory must maintain a list of tests with critical values. What will be the critical values? Very high glucose, very low glucose, very high creatinine, very low creatinine, very low platelet, very high total leukocyte count, any positive microbiological culture with certain specific organisms, any specific malignancies detected in your laboratory. So make a list of the tests with their critical values. Many of the time there may not be any critical values for any of the tests. So leave it. But what are the common tests? What are the tests where the critical values are applicable? You must mention, you must prepare a list of all those critical values. They should provide critical values to clinicians on telephone. So it is expected that all critical values must be informed to the clinicians by telephone or in any other way which is reaching to the clinicians or the concerned healthcare personnel as early as possible. So glucose values of 45, here are some examples they are mentioned. So I have taken those examples itself. Glucose value of 45 milligram, frozen section biopsy report is required when reporting a patient with suspicion of cancer, growth of a particular organism in culture for early diagnosis and treatment. So all this will come under critical results, and you should record those all critical results whenever you are getting such result in your laboratory, and it should be immediately informed to the clinician. Now coming to the automatic release of reports can be applied if the laboratory has necessary alliance or middleware capabilities and has competency to implement it. So many a times nowadays we have seen multiple softwares, alliance solutions, middlewares have come which have a capacities to automatically select and release the test report based on the predefined criteria or based upon the predefined validated criteria. So you must have a validated criteria so that all those reports can be validated as per the specific algorithm which is defined in your LIS or in your middleware, and report will be automatically released. So it reduces the lab work. Many times we get thousands and thousands of tests per day. So it is not possible to release all or to go through all the test results, even if they are now all the values are within normal reference range, within biological reference range. It is not possible to go through all those results. So what you can do, you can use certain algorithms as per your capacity. Here it is mentioned as per the competency. So as per the competency of your LIS, as per the competency of your middleware, and as per the competency of the personnel who are defining those algorithms. So as per the competencies, you can implement this automated release of reports by your LIS and by your middleware. Now coming to the post-analysis part in sample storage. So storage of samples, whenever you are storing any sample, like routinely we store samples for a specific period of time. After the analysis is performed or after the examination is performed, for a specific duration, the samples are still retained in the laboratory. So what are the things which is prerequisite for required conditions, under which conditions you are going to retain those specimens? Under frozen conditions, under 2 degrees Celsius, under room temperature. So you should know specific conditions, method of storage, stability of analyte. Even after storing under specific condition, how many hours it will be stable? What is your retention period for each and every test and each and every specimens should be followed as per the requirement of the users and as applicable national regulatory guidelines. So during COVID, most of you might have seen previously the guidelines were you should keep the samples for 1 year, but when the volumes of samples increased to thousands and lakhs, then government has given you relaxation from your corporations and your health authorities. They give you relaxation that negative samples can be discarded within 7 days. Positive samples, their RNA should be retained by the laboratory for 6 months or 1 year. So those guidelines keep changing. So as per the national and regulatory guidelines, you should retain the specimens for a defined time period under specific conditions so that the stability and integrity of the analyte can be maintained. Every effort should be made to retain blocks and slice in the histopathology and cytopathology. So blocks and slice of histopathology and cytopathology should be retained for as long as they are relevant, or if there are any specific accreditations or regulatory guidelines. Maybe some guidelines mention it to keep for 10 years, some guidelines may mention it to keep it for 5 years. So please check what type of specimens you have, what are the applicable guidelines with respect to those specimens. As per the guidelines, as per the regulatory agency guidelines and national guidelines, you have to retain those blocks and tissues till the retention period or for a minimum retention period. You can maintain, if some guideline is mentioning that it should be maintained for 10 years, and if you want to retain it for 15 years, 20 years, it's up to you. But minimum 10 years it should be retained. Now many a time our routine hematopathology and biochemistry specimens are required to be maintained till 24 hours. Now, if you decide and if you have sufficient conditions to store, if you want to retain them for 72 hours or even for 7 days, it's up to you if you are able to properly retain them and if there is any specific purpose to retain them. Okay, so it's up to you, but a minimum retention period must be followed, whichever is as per the guidelines. If, however, they are returned to the patient, this must be documented and records must be maintained. Now, many times blocks and slides in histopathology and cytopathology are returned to the patient. So, in that case, you must have appropriate documentation and records like whatever requisition the patient has given, asking for the returning of those blocks and slides, requisition from patient. If there are any appropriate documentation, you should have your records. You should take counter signature from the patient. So whatever is applicable documentation is there, it should be maintained if you are returning the specimens, blocks or slides back to the patient. Appropriate, because it is for your safe, or if for in future if something or some dispute arise, you should have appropriate documentation with you regarding the return of blocks and slides. Samples after retention period, if any, should be segregated in color-coded bags at source or in sharps container as applicable. So whatever type of samples you have, all those must be segregated as per the color-coded bags. For this, there is no other document other than the biomedical waste guidelines. So you have to follow whatever is given in the Indian biomedical waste guidelines as per the guidelines of 2016 or 2018, whichever is applicable. After that, during COVID, as I have mentioned, there were certain amendments regarding the segregation of the waste. So those criteria must be followed for discarding of your specimens. This should be handed over to approved local biomedical waste disposal agency. So there should be a appropriate MOU with the biomedical waste agencies which are approved, which are authorized. To them only you should be able to handle the waste. Now many a times from my experience, I have seen there are many small labs. So what they will do, they will have certain 3-4 friends who are running the lab. Only one person will sign the MOU with the biomedical waste agencies. All of those 4 labs will collect the waste and they will hand it over to a biomedical waste disposal agencies. So it becomes a certain unauthorized or it becomes certain unregulatory disposal of your biomedical waste. So for this purpose, and it doesn't cost much. So you should have appropriate biomedical waste disposal agreement with the authorized agency only. Do not try this type of small cost saving approaches, because ultimately it will hazard to the public or it may cause hazard even to your staff or yourself itself in your laboratory. So have a proper biomedical waste disposal agreement. Care should be taken in disposal of hazardous chemicals, if any, used by the laboratory. Now, disposal of hazardous chemicals, we are using hypochlorite, we are using certain acids, alkalis, we are using this formalin, xylene. So all those must be appropriately treated and appropriately disposed of. Like I think on the first day there was one question like how to discard this xylene and formalin. So first of all, you have to treat it with hypochlorite. Then it should be going to appropriate STP plant, SGT plant, and then maybe it can be discarded in a public waste. And you should have approvals for all those facilities of STPs and hypochlorite addition facilities and appropriate biomedical waste discarding and storage activities. And you should have those plant, STP plant, if you are going to discard it, or maybe sometimes you can just return that formalin, whichever is used, to the back to the manufacturer. If you know he is going to take care of it properly and he is going to recycle it, or if they have appropriate documentation of recycling of this formalin, so you can discard it in that way also. care should be taken in disposal of computer hardware and other items. So maybe you will have certain condemned hardware and certain condemned electronic items. Mobiles will be there in the laboratory. Certain other phones, other aspect will be there. E-waste will be there. So all these e-waste also should be appropriately discarded in your laboratory as per their guidelines. So there are guidelines like how you have to recycle it or to whom you should hand it over properly. All those precautions must be taken while discarding the e-waste also. So with this, we are able to close this today's session. So if you have any doubts, we can take it now. Measuring range is also mentioned. Backup, see, there is nothing like backup machine and main machine. All the things you have to do for all the machines, wherever, whether it is a backup machine or the main machine you are using, they should have appropriate measuring range. They should have appropriate accuracy and precision. Okay. So basically you have to do it, whatever equipment is available in your laboratory and whatever measurements you are doing in your laboratory, whichever test method you are doing, it should be available. How many samples in minimum are required for verification of a test? So there are various protocols. The standard protocol says 20 samples for precision, 2 duplicates in accuracy. Then there are certain protocols for carryover studies. So all those protocols must be followed by the laboratory. Please ask your manufacturer. They have their specific protocols, or you can take these protocols from any of the authorized textbook. Whatever textbook we have studied in our graduation, post graduation, every textbook has this protocol of verification, because I do get these questions many a time, and there are various papers and publications. So take a proper reference, use those protocols for verifications of your examination methods in your laboratory. How to perform validations? So validations also, there are a lot of protocols. How to come to conclusion of accuracy, how to come conclusion of precision, how much sample size is needed. Maybe you can take a guidelines from whatever has been given in your kit manufacturer literature, how many samples they have used for validation. You can take it as a surrogate data to modify your modification, to modify your test methodologies. You can do a sample size calculation. So basically, I'll tell you, validation is a part of research. So you should know appropriate protocols when you are performing any research, how many samples you have to perform, how much should be the sample size. All those aspects comes into the picture. Apart from that, many ethical considerations also come. So you have to look into all those aspects whenever you are going for any validation. Sample type beyond manufacturer's instructions, I have mentioned. You can take any protocols, or you can even ask, or you can take a surrogate protocol which your manufacturer have already used for the recommended specimen. The same protocol you can take for extending the use for any other specimen. What is the procedure for verifications? I have already mentioned that. I can tell you the characteristics which you have to verify, like precision you have to verify, accuracy you have to verify, linearity you have to check. You have to check biological reference interval, you have to check for interferences, and the protocols are mostly like 20 samples you have to run for each aspect. This is a rough protocol I have given. Apart from that, then there are various small, like there are variations in that, like in precision, if you run 20 samples, you have to do 4 samples for 5 days in a line. So that we call it as a protocol of 4 into 5 for precision. We call it as a 4 into 5. For accuracy, we use 2 samples or 2 reference materials which has accurate values of higher level and low level. Those should be run in duplicate. Apart from that, in your interference studies and in your biological reference interval, you should have 20 samples should be run for your verification of biological reference interval. Various interferences can be studied using spiking of the sample. So there are various protocols. We can use those protocols for your validations and verification. Should the biological reference range be that of the local population? Yes, if it is available for your local population, like if you are living in certain part of India or certain part of the world, and you know there are reference ranges which are already established, then you have to use that, and it is more relevant for your population. In India, for now, ICMR has taken up a very major project of verifications of reference intervals for all the examination processes and test results in different parts of the India. and soon we will be having reference intervals for all the populations in different geographical locations in India. So you can use that whenever the data get published. QC run for backup equipment, as I have already mentioned, there is nothing like backup and main. Whenever if you are running the samples, you should process the QC. Whenever you are running the samples, see if you are running some samples today, if you are not running it tomorrow, today if you are running the samples, you have to run the QC. Tomorrow if you are not running the samples, there is no need of running the QC. Okay. As per NABL 112 or as per those guidelines, they may have different guidelines, or they will have some specific guidelines. If they have specific guidelines, you have to follow them. If there are no specific guidelines, for your purpose, you can do appropriate risk analysis of running the QCs and you can define your protocol. For simple purpose, if you are running the samples, you need the QC. Without running the QC, you cannot run the samples, whether it is a main machine or backup machine. Which reference interval is correct, hit or universal for any test in pathology? Please suggest whichever is relevant to your population is a correct reference range. Okay. Apart from that, if you are taking it from some kit or if you are taking it from some universal international textbook, see whether your clinicians are comfortable with that, because they are the consumers of your test results. Whatever results are generated by your laboratory, your clinicians, your population is the consumer. So those reference range should be suitable to them. So please check from wherever you are taking the reference interval, it is suitable. It should be suitable for your population. Can we report values beyond linearity range? Yes, anytime. If you have verified the reportable range, you can report the values beyond your linearity. For CRR, as I have mentioned, there are references that at least 20 samples from all the study populations you should run, and out of 20, 19 should fall within your defined reference range. When we run QC in the morning and result comes either 1 SD or minus what, the difference is 10% from mean. In that situation, our also result comes with the same difference according to control or not. Yes, it can come. See, it is nothing like a mathematics. If here you are adding 1, there also should add 1. It is nothing like that. There are certain variations you have to consider those variations. QC material is not exactly the samples which is related to the patient blood. There are certain preservatives. Stability of the QC sample varies. They have been manufactured long ago, maybe 6 months or 8 months before. Till they are reaching your laboratory, they might have undergone various environmental conditions. So we cannot guarantee that if you are adding plus 10% there, the same 10% will add to your patient sample also. Okay. So you have to take other factors also into consideration while you are reviewing the QC results. In histopathology reporting, is the timing of reporting important because we practice the tissue in batches irrespective? It may not be important for all histopathological patients, but if you are reporting something like frozen section, the report needs to be completed within 35 or 30 minutes because the patient is already on the table. So in that case, the timing is important. For other histopathological examinations, timing may not be important, but again, to calculate the TAT, to calculate other operational parameters in your laboratory, you may have to record the time of reporting also. Sometimes we see our control results are not under the mark, but we repeat our last test samples and results comes as yesterday when our control results was under the range. So what is the mean of control? So it is up to you how to interpret. That means your control itself is not stable. If your patient values comes the same, but the control values are not within the range, that means your control itself is not in the stability period. It has lost the stability. So that means you are not storing the controls appropriately, you are not reconstituting the control appropriately. The run protocols which you are following for running the controls itself is not proper. Okay, so controls are meant to review your reports in a real-time basis, that whatever reports are released by your laboratory are reliable. So if you follow the correct protocol, the controls will give absolutely correct result. If the sample is diluted, should it be reported in the test? Yes, you can mention it. It's up to you. It's up to you. If you are running the samples in dilution, you can mention it that the sample was run in 10 dilution or 15 dilution. Lab director delegation of duties in presence she or he assigns senior biochemist assigned duties for monitor of the QC. Like that, see, monitoring of the QC is the supervisory personnel of the respective department. Not one lab director sit and can monitor QCs of all the areas. Maybe one director is there and he has under 10 or 15 areas under him, and each area is performing thousands of tests per day and they are running hundreds of QCs per day. It is not possible, practically possible for one person to monitor everything. So they may assign duties to somebody else. It's up to your laboratory protocol. Difference between amendment and record report, nothing is there. Amendments means some results you have to change, or some results you may have to edit them. So for that you have to recall them. Without recall you cannot amend them. No? So it is basically recall. After before amending you have to recall them, then only you can amend those results. Is the amendment procedure form exists? Amendment procedure form you can make as a standard SOP in your manual, or you can put it as an annexure. So it's up to you. There is no hard and fast guidelines that you should have appropriate SOPs, you should include it. It's up to you. What must be contained the reported original? And we have given one format, so you can see the format. What are the different content should be there for appropriate report? Do we need to report a critical value for a known critical case? If it is relevant, if your clinician wants you to report all the critical values, then you have to report, because ultimately it is for the users. You must able to meet the requirement of the user. So if your users want that all critical values, irrespective of the criticality of the case, they want to them to be informed, inform them. Any standard to create algorithm, you can use multiple, like I can say you can use multiple criteria, like sample results falling within reference range, samples which are passing delta check, samples which are correlating clinically. All those protocols you can include in your algorithms for validation of the results in auto verification. Should we treat blood and other body fluids? Potentially, all body fluids and bloods are the parts of the biomedical waste and should be handed over together. And why you want to segregate it? It's all waste, biomedical waste, and body parts or body fluids. Blood itself is a body fluid. Okay, so you can hand it over together. Should we treat? Should we give amended report? Should we keep the original report in the same report? You have to keep records of the original report as well as amended report. Both you have to keep. Within run, between run. Within run means in one run, same sample can be repeated twice. Between run means one sample in two different runs can be repeated. Verification methods I have included in a very short. What are acceptable difference between two machines with same method? So if both the machines are with the same methodologies and with same manufacturer, ideally there should not be any difference. Okay. How to calculate MU? As a simple mathematical calculation, 2 into CV percentage. 2 multiplied by your CV percentage should be your measurement uncertainty. This is a simple calculation which can be used. Reference ranges, again I have mentioned that it can be taken from any textbook which is relevant to your population. Do we mention substances not interfere? Yes, we have to mention in your SOPs substances which are interfering in examination. Formalin monitor requires calibration. Absolutely, it is an analytical equipment which measures something. So anything which is measuring any quantity, anything which is which has anything measurable component should be calibrated. What exactly should be written in an amended report? Who has amended the report? What was the reason for amendment? What is the date and time of amendment? Everything should be captured in your amended report. Is it mandatory to separate the serum in vials for storage after analysis, or it can be stored as such in a vacuum? It depends upon what type of vacuum and how you are storing it. Once you have centrifuged the sample, the serum is separated from the other cells. So you can store it as it is, provided that the stability remains and there are no further instability in your samples, or if your not samples are appropriately vertically stored in your storage conditions. Fluid size, how we have to save for homogeneous cytopathology, I have mentioned already. As per some guidelines, it is for 10 years. You can check other relevant guidelines whenever it is required, but as far as possible, it should be for 10 years. If two machines alternatively use, yes, you have to do inter-machine validation. Of course, if you have more than 2 or 3 machines for the same test, irrespective of the use, how you are using, you have to perform the inter-machine comparison. Reference ranges can be validation should be done by the laboratory itself. In a microsite lab, it is less than 20-30 years, but it is 24 hours. And what should be the QC frequency? So if you are running the Q samples in all the shifts, you need to have appropriate risk-based QC frequency should be defined. If there are any regulatory guidelines, if there are any guidelines for accreditations, you have to follow that. If there are no guidelines from any accreditation bodies or from any regulatory bodies, you can define the QC frequency depending upon the risk analysis in the QC. You can define it by yourself, absolutely. If provided that there are no guidelines, but if there are guidelines, you have to follow the guidelines. There is no other way. How often inter-machine comparison should be done? Maybe once in a 6 month or once in a year will be sufficient. If IQC should be calculated, of course, CV you have to calculate total error. You will not be able to calculate from just IQC. Is it mandatory to run both level of QC? Yes. If you see the kit inserts of all the manufacturers, they recommend running minimum 2 levels of QC. So there is no alternatives because whatever manufacturers guidelines are given, you have to follow. There is no alternative. How to dispose of residual urine? You should dispose of it appropriately, considering it as a biological waste from the human body. Guidelines are applicable because it is an infectious waste. Okay. How is spatial integrity checked? Spatial integrity can be checked by repeating the same samples on day 1, day 2, day 3 as till the retention period. Is lab to be derived from every lot? Yes, lab needs to be derived from every lot as far as biochemistry is considered. For hematology, it may not be required because your lot changes every 15 days or every month. So it will not be practically feasible to arrive at a laboratory lab developed mean. What about 3 level of QC versus 2 level of QC? Many times your values may fall on lower side or in the within reference range and at higher than reference range. So to cover all the reference ranges of low, mid and high, you should have QC values spreading across all the 3 levels. So if it is available, you have to use it. In actual lab, creatinine reference is 0.95 to 95 for female Japanese, but most of the patients values coming below 5. Please check your examination method which you are using or how the procedures are actually performed. Is it mandatory to display the contact number of application person in your instrument? Information should be available. Okay, information should be available. Whether you have to keep it near the equipment or away from the equipment, it's up to you. If patient from very cold place, Siberia, Uzbekistan, anywhere, then our PT control analysis will be same for them or not. No, it will vary. So as I have mentioned, your PT INR values will depend upon your MLPT. So whenever you are receiving a new lot of prothrombin and activated prothrombin reagent in your laboratory, you have to perform the MLPT for every lot. Depending upon that, your PT controls and INR value will differ. Okay, so whichever part of the country you are going, you have to first establish PT controls and INR values with the help of MLPT. How we discard polyamine and xamine? I have already mentioned that you have to use appropriate hypochlorite. You have to do appropriate sewage treatment plant should be there, and then only you can discard it to the public waste. Who can amend the report? Anybody who is authorized. Anybody to whom your laboratory has given authorization. Okay, they can amend the report. No, you need not have to mention substances not interfering in the SOPs. How many samples should be done for inter-machine comparison? Again, 20 samples will be sufficient. Again, this is as per some guidelines. The guidelines may vary for equipments and parameters. Yes, you can reject the samples if it is not possible. If he is blocking the flow, So you should be knowing what type of probe samples your laboratory will be able to analyze. If your laboratory is not able to analyze such viscous samples, please don't accept those samples itself. You can just reject it. You need not have to accept all the samples and run all the tests. Can autopsy come under scope of enable accreditation? Absolutely no. It is for the. Thank you for the presentation. Sample integration of retention sample. What is the frequency it should be done? It can be done once in a month. Okay. Sample retention study. How can we get MNPT only for one patient from outside? See, in that case, your reference ranges will not be applicable. Okay, if you are getting one patient, then it's you can discuss with the patient and you can tell them that these reference ranges are not applicable. For one patient, you cannot do MNPT and PT. You are doing it for your laboratory or with the population which is based geographical population which is based in your area. So if somebody once in a year or once in a while is coming from some other part of the country, you can always discuss with the patient or the clinician and can tell them this population may not be applicable to them. So if you want any additional sessions, because I can see there are some requests for some additional sessions for LJ chart validations, verifications. If you have any such request, please forward it to the Mindray so that they can arrange it, or we can take it back. I think Nikhil is there.
Yeah, yes, sir, I'm here.
Maybe we have already circulated the feedback form. So if you have any additional request,
Definitely.
and if we receive some sufficient number of requests, then we will surely take those additional topics.
Definitely, sir. Definitely, we'll compile and we will.
Yeah. So I think
yes,
I have tried to answer all the questions, and maybe we can.
Sir, one from other platform also. He is asking. Dr. Reena is asking, is AMR verification needs to be done for hematology? And she's asking, is linearity study and AMR verification the same?
See, AMR validations and linearity study, and you have specifically asked these questions with respect to the hematology. In hematology, this part is taken care during the calibration itself. So you need not have to do any independent AMR verifications because it is a part of calibration for hematology, whether it is a 3-part counter or whether it is a 5-part, 8-part counter. The calibration parts take care of all these activities as far as hematology is there. So if it is for biochemistry, absolutely you have to do it separately because the calibration doesn't take care of all those activities. But if it is a hematology, if you see the calibration records of your equipment, you will see it has been already done during the process of calibration. So you need not have to do it separately anything.
Thank you, sir. One more question, sir. Dr. Jadhav is asking, where ETP is not mandatory or not available, how to dispose of the liquid waste?
So for that purpose, like I can see that these questions may be some small labs or some standalone labs where which do not have this STP plants. So please communicate or talk with your biomedical waste collecting agencies whether they have any such procedures or they can carry it separately because they are having this disposal plant and STP plant. Ask them, request them, they may charge you separately something, but it is better you talk with them, make a separate amendment or make a separate MOU with them how this liquid waste will be carried. So they will help you better. Many times I have mentioned the supplier itself will take it back for recycling. There are various protocols available for recycling. So if your supplier is taking it back and recycling it, provided they have appropriate licenses for that. Also, they should also have appropriate licenses for recycling. So check that you can return it back the use formalin, you can collect it at the end of the day and you can return it back to the supplier itself, or you can hand it over to the biomedical waste collecting agencies. But don't hand it over directly. Please talk to them what type of protocols they have, how they are going to discard it, whether they have appropriate licenses for discarding it. Then only you can hand it over to them. Okay.
Thank you, sir. Sir, one
There
last.
are a few hands raised, and I can see Dr. Mangala Nagre. She has raised her
Whoever
hand.
has raised the hands can I
Can
have given the
unmute
mic.
and
Yes,
yeah,
they can unmute
please
and
unmute and ask your question. So we still have 2 minutes.
Yeah. One by one, if they want, they can unmute themselves and ask.
Yeah. So please unmute and ask, Dr. Mangala, please unmute and ask your question. Mr. Bapurao Kale. So it seems they have raised their hands and gone away. Mr. Mubarak Basha has unmuted himself. Yeah, Mr. Mubarak Basha, you can speak.
Yeah, we have given you speaking right, so you can just unmute
Are
yourself.
they able to speak, Nikhil?
Yes. Those all the photos you see on the screen, they are allowed. That means I have given them the made them as
Permission.
yeah,
Okay.
yeah.
Okay.
So anybody who wants to quickly clarify
I
that,
don't think Mr. Mubarak Basha has unmuted himself, but
maybe
has not able to. I think he's not able to talk.
anybody anyway, no problem, sir. We can then maybe close it and then come back.
Yeah, probably. So thank you everyone for joining today, and it was really a good session, very interactive, and lots to learn from Dr. Jayesh Warade still. So tomorrow, same time, we join at 7 p.m. again and continue with this webinar series. Thank you, Dr. Jayesh, for
Thank you.
this
Thank you.
enlightening talk. Thank you, Nikhil and Namrata,
Thank you.
for the coordination. Thank you. And tomorrow we meet at the same time, 7 p.m. Thank you.
Thank
One
you.
one
Thank you,
small
everyone.
request, one small request. When you close the webinar, you will get a feedback link. So if you have any suggestions, if you have any request, you please let us know. We will compile it, and whatever the
Yeah.
maximum number of queries we have received, we will try to pass it on to sir and arrange for the next session. So please do help us with the rating and your suggestions also. Thank you so much. Thank you.
Thank you. Thank you.
Thank you, everyone. Bye-bye.
Thank you, everyone. Bye.